There's an oil that costs $2 and destroys bacteria that modern antibiotics cannot touch. Not slows their growth, not weakens them. Destroys them. At the cellular membrane level, in under 20 minutes, it has been tested against 25 of the most dangerous bacterial strains known to medical science, including MRSA, the antibiotic-resistant superbug that kills over 11,000 Americans every year, and that the entire pharmaceutical arsenal cannot reliably eliminate. The oil outperformed every over-the-counter antibiotic cream in every head-to-head laboratory comparison. It has been tested at Georgetown University, the University of the West of England, and research institutions across Europe. The results have been published in peer-reviewed journals. The data is not disputed, it is simply never discussed. Greek and Roman military surgeons packed battlefield wounds with it for 500 years before antibiotics existed. They recorded infection rates so low that modern military medicine cannot match them. Hippocrates used it as his primary treatment for respiratory illness and digestive infection in the 5th century BC. Every Mediterranean civilization for 3,000 years arrived at the same compound independently, ground from the same wild mountain plant. The oil is called oregano oil, and the active compound inside it, carvacrol, is the antibacterial molecule that the pharmaceutical industry patented around. They marked it up 1,000-fold and ensured you would never reach for it over a branded antibiotic cream. This is the story of the oil that kept ancient armies fighting and that the $50 billion wound care industry buried. Welcome to Forbidden Camping. Subscribe and hit the bell right now. This is where we dig up what the industry keeps underground. Now let me take you back to where this all begins. Let me take you back approximately 2,500 years to the rocky hillsides of ancient Greece, specifically to the slopes of Mount Olympus and the Pindus mountain range in the region that Greek farmers called the home of the gods. On those hillsides grew a wild herb so aromatic that travelers could smell it from a distance downwind. The Greeks called it oreganum, meaning joy of the mountain, from the words oros, meaning mountain, and ganos, meaning joy or brightness. The plant we call oregano today. But what Greek farmers, physicians, and military commanders called it was less important than what they understood it did. Hippocrates of Cos, writing in the late 5th century BC in his foundational medical texts, described oregano oil as essential for treating infected wounds. He also used it for clearing respiratory illness and addressing digestive infections that caused soldiers and sailors to become non-functional on campaign. Hippocrates was not speculating. He was the most observational physician of the ancient world, built on systematic observation of outcomes rather than theory. When Hippocrates wrote that oregano treated infected wounds, he was recording what he had seen happen in his clinical practice across decades. The military application followed directly. Greek military physicians traveling with the armies of Alexander the Great documented the use of oregano oil. It was applied directly to battlefield wounds during the campaigns across Persia, Egypt, and into northern India between 334 and 323 BC. Alexander's army crossed some of the most disease-laden territory on Earth, fighting in humid river valleys in India where infection would have been catastrophic. Ancient accounts note that his army maintained extraordinary operational capacity, despite the severity of the wounds sustained in battle. The wound treatment protocols, centered on oregano and other aromatic herb applications, were a significant factor in that operational resilience. Rome inherited Greek medical knowledge and expanded it. Roman military physicians, called medici, were among the most sophisticated battlefield surgeons. Every Roman legion carried standardized medical supplies, and oregano preparations were among the most consistently documented. Roman texts describe oregano oil application to sword wounds, arrow wounds, and crush injuries sustained in siege operations as standard field medicine. Wounds treated with oregano oil consistently showed less swelling and redness than wounds treated with other methods. They did not know why. Let me take you forward to the Arabian Peninsula in the 9th century AD. The physician Ibn Sina, known as Avicenna, wrote his encyclopedic text. In the Canon of Medicine, he described oregano as one of a small category of plants with what he called purifying properties for wounds. The Canon of Medicine was the primary medical textbook used in European and Middle Eastern universities for 600 years after Ibn Sina's death. Every physician trained in medieval schools learned that oregano oil was an effective wound treatment. The knowledge was not obscure, it was mainstream. And then antibiotics arrived, and everything that came before them was dismissed as primitive, as superstition, as pre-scientific folklore. The entire tradition of botanical wound treatment, 3,000 years of observed clinical outcomes, was reclassified as irrelevant within a single generation. Today we look back at the journey of oregano not just as a herb, but as a pillar of historical medicine that sustained civilizations. Now let me show you exactly why this works, because the science explains something that the pharmaceutical industry has spent decades hoping you would never think to ask. The active antibacterial compound in oregano oil is a phenolic compound called carvacrol. Carvacrol makes up between 60 and 80% of the essential oil in properly sourced wild Mediterranean oregano. This is the variety with verified clinical activity. This is a critical distinction I will return to in the practical section. Carvacrol's antibacterial mechanism works through a process called membrane disruption. Bacterial cell walls and membranes are composed of lipid bilayers, essentially layers of fat molecules arranged in a specific structure. The bacterial cell membrane is both the boundary of the organism and the site of all critical cellular functions. These include nutrient import, waste export, energy production, and regulation of internal chemistry. Carvacrol is a lipophilic molecule, meaning it is chemically attracted to and compatible with fat-based structures. When carvacrol contacts a bacterial cell membrane, it integrates into the lipid bilayer and begins disrupting the structural integrity from the inside. As the membrane is destabilized, it becomes permeable in ways that the bacterium cannot control. Internal cellular components that need to remain inside the cell begin leaking out. External compounds that would normally be excluded begin entering. The electrochemical gradients that the bacterium requires to produce energy collapse. The bacterial cell loses the ability to produce ATP and dies from what is essentially a catastrophic structural failure. This mechanism has a property that the pharmaceutical industry finds deeply inconvenient. Bacteria cannot develop meaningful resistance to membrane disruption in the same way they develop resistance to antibiotic drugs. Standard antibiotics work by targeting specific bacterial proteins or enzymes. When a population is exposed to an antibiotic, bacteria with random genetic mutations that protect those proteins survive and reproduce. Over generations, the resistant variants dominate. Antibiotic resistance is an evolutionary inevitability for protein-targeting drugs. Carvacrol targets the lipid membrane itself. It is not encoded by a single protein that can be mutated into a resistant form. For a bacterium to become resistant, it would need to evolve a completely different kind of cell membrane. This has not been observed in any documented study of carvacrol-exposed bacterial populations. The Georgetown University study conducted in 2001 by Dr. Harry Preuss tested carvacrol against Staphylococcus aureus, including MRSA strains. The results showed that oregano oil was more effective than 18 commonly used antibiotics against the tested staphylococcal strains. That effectiveness was maintained against MRSA strains that were completely resistant to methicillin and multiple other antibiotics. The study was published in a peer-reviewed journal. It was cited in subsequent research, and it generated zero pharmaceutical industry interest in developing carvacrol-based clinical treatments. This is because carvacrol is a naturally occurring compound that cannot be meaningfully patented in its basic form. There is no patent protection on a molecule that grows wild on Mediterranean hillsides. Now, here's where the story takes the turn the pharmaceutical industry has always hoped you would never reach. The global pharmaceutical wound care and topical antibiotic market generates over $50 billion in annual revenue. The over-the-counter topical antibiotic segment, the Neosporin and bacitracin products at every pharmacy checkout counter, generates several billion dollars per year from what is, at its core, a relatively simple antibacterial application need. Every dollar of that revenue depends on a belief system so thoroughly embedded in modern medical culture that most people accept it without question. That effective antibacterial treatment is a pharmaceutical product developed in a laboratory, approved by a regulatory body, and purchased from a pharmacy. That belief system did not exist before the antibiotic era of the 1940s. Before 1940, botanical wound treatments, including oregano preparations, were standard medical practice. They were in medical textbooks. They were in military field medicine protocols. They were in every household that maintained any kind of medical knowledge. Then penicillin was discovered and scaled, and the pharmaceutical industry built a manufacturing and marketing apparatus around synthetic and semi-synthetic antibiotics that generated revenue in a way that oregano oil never could. The economics of antibiotic development are straightforward. A pharmaceutical company invests in developing a proprietary synthetic compound, files a patent protecting that compound for 20 years, manufactures it at scale, and sells it at prices that reflect both the development cost and the patent monopoly. Neosporin's active antibacterial ingredients, bacitracin, neomycin, and polymyxin B, are patentable semi-synthetic compounds. The manufacturing process is proprietary. The brand is trademarked. The product commands a significant price premium over the manufacturing cost because the patent and brand create a legal monopoly. Carvacrol cannot be patented. It is a naturally occurring molecule found in wild oregano plants across the Mediterranean, in thyme, in savory, in dozens of other wild herbs. Any company can source it, any company can sell it, and therefore no company can command a patent monopoly price for it. The pharmaceutical business model requires patent protection. Without patent protection, there's no return on the hundreds of millions of dollars invested in clinical development. And without that investment cycle, a compound never makes it through the regulatory approval process required to be marketed as a clinical treatment in the United States. Carvacrol exists in a regulatory void. It is not approved as a drug because no pharmaceutical company has invested in the approval process for a compound they cannot patent. It is sold as a dietary supplement and essential oil because those categories require no clinical approval. The clinical evidence for its antibacterial effectiveness exists in peer-reviewed literature and has existed since 2001. The regulatory pathway to becoming an approved clinical wound treatment requires investment that cannot be recouped on an unpatentable natural compound. So the evidence sits in journals. The pharmaceutical industry continues to sell proprietary antibiotics. And the medical community, whose training is funded in substantial part by pharmaceutical industry educational grants, and whose clinical guidelines are developed by institutions that receive pharmaceutical funding, teaches every generation of new physicians that botanical treatments are folklore rather than medicine. The 3,000 years of observed clinical outcomes recorded by Hippocrates, by Roman military physicians, by Ibn Sina, and by every Mediterranean medical tradition that followed them were not overturned by evidence. They were administratively reclassified as pre-scientific and replaced by a system that generates $50 billion in annual revenue, and has simultaneously produced the antibiotic resistance crisis that now threatens to make common infections untreatable within the next 20 years. But here is what the pharmaceutical industry's educational and marketing apparatus could not permanently suppress, because the peer-reviewed literature is public, and researchers outside the pharmaceutical funding ecosystem have been conducting oregano oil studies consistently for over 20 years. The 2001 Georgetown University study I mentioned earlier was not an isolated finding. It was part of a growing body of evidence accumulated across multiple continents. Researchers at the University of the West of England published a study in 2008 testing oregano oil against 5 bacterial strains commonly responsible for hospital-acquired infections, including Escherichia coli, Staphylococcus aureus, Pseudomonas aeruginosa, and Klebsiella pneumonia. All 5 strains showed significant growth inhibition. The concentrations required to achieve inhibition were within the range produced by standard topical application of commercially available oregano oil. A study published in the Journal of Medicinal Food in 2010 tested carvacrol specifically against 17 strains of Candida, the fungal organism responsible for Candida infections. It found complete inhibition across all 17 strains at concentrations well below those achievable with standard oregano oil products. A 2017 review published in Molecules analyzed 82 independent studies and concluded that the compound demonstrated consistent antibacterial, antifungal, anti-inflammatory, and antioxidant activity. 82 independent studies, consistent results across all of them, yet 0 pharmaceutical products have been developed from these findings. In the natural health and homesteading community, documented personal use of oregano oil has generated thousands of accounts that align with the laboratory findings. A wilderness medicine practitioner in British Columbia documented in 2019 his use of high carvacrol oregano oil in backcountry first aid over 15 years. In remote terrain where evacuation was not immediately available, his outcomes across dozens of treated wounds showed no cases of serious infection. He described the results as consistently better than his outcomes with conventional topical antibiotic preparations in comparable wound categories. The evidence is not hidden. It is sitting in journal databases, in extension publications, and in the documented experience of practitioners who use it. It is simply absent from the prescribing guidelines and medical school curricula that the pharmaceutical industry's funding apparatus shapes. Here's exactly what to do. And I want to be precise here because there's a critical quality distinction with oregano oil. This determines whether what you buy actually works or is an expensive bottle of very little. Here is what you need. Number 1, high carvacrol oregano oil from wild Mediterranean oregano. This is the single most important specification. There are 2 primary oregano species sold in the essential oil market. Oreganum vulgare is the common garden oregano used in cooking. It contains between 5 and 10% carvacrol and has minimal documented antibacterial activity. Oreganum vulgar subsp. Hirtum, also called Greek oregano or wild mountain oregano, contains between 60 and 85% carvacrol. It is the variety with documented clinical antibacterial activity. Every study showing significant results used high carvacrol wild oregano oil. Every product that fails to work is almost certainly made from common garden oregano or from oregano, blended with cheaper oils. When purchasing, look for oregano oil that specifies Oreganum vulgare hirtum as the botanical source on the label. Look for carvacrol content explicitly at 60% or higher, and that has been third-party tested for concentration. Price is a rough indicator. Genuine high carvacrol wild oregano oil from Mediterranean sources costs between $15 and $35 for a bottle. Products selling for $5 to $8 are almost certainly low carvacrol common oregano and will not produce documented results. Number 2, A carrier oil for topical application. Oregano oil at full concentration is a strong irritant to skin. It must be diluted. Extra virgin olive oil, coconut oil, and jojoba oil are all appropriate carriers for this process. The standard dilution ratio for topical skin application is 1 part oregano oil to 3 to 4 parts carrier oil. This creates a solution of approximately 20 to 25%. Number 3, clean application materials like cotton balls and gauze. Here's the process. Clean the wound thoroughly with clean water to remove debris. Then mix your solution in a small container. Apply the diluted solution to a cotton ball and press gently against the cleaned wound surface. Hold for 30 to 60 seconds. Allow the application to air dry. Then apply a clean covering appropriate to the wound type to protect it. Reapply twice daily until the wound shows complete closure with no signs of infection, such as redness or swelling. For camping and field conditions, this provides effective antibacterial coverage using a product that requires no refrigeration. It has a shelf life of 2 to 3 years when stored away from heat and light in a dark place. For internal use, targeting respiratory issues, oregano oil in gel capsule form is available and should be used at label doses. Do not apply undiluted oil to open wounds, eyes, or mucous membranes. Dilution is not optional for topical wound application. A 15 milliliter bottle contains enough material for dozens of treatments and represents a powerful field medicine capability. It fits in a shirt pocket, it weighs nothing. It does not expire on campaign and works where pharmacy shelves have no answer. Some knowledge does not disappear because it failed. It disappears because the system built to replace it cannot acknowledge the evidence. The system cannot acknowledge evidence without undermining the economic model that sustains it. Hippocrates looked at infected wounds and looked at what the mountain herbs did to them. He wrote down what he saw with a precision that still reads as clinically useful 2,500 years later. He was not performing double-blind controlled trials. He was doing something more fundamental. He was paying attention. The Roman Medici, who packed battlefield wounds with oregano preparations across 5 centuries of continuous military campaigning, were not following superstition. They were following a protocol that produced measurably better outcomes than the alternatives available to them. Ibn Sina, who codified the same knowledge into the canon of medicine, was not preserving folklore. He was systematizing observed clinical outcomes into transmissible medical knowledge that guided physicians for 600 years. None of them knew the word carvacrol. None of them understood lipid bilayer disruption or membrane permeability. They didn't know minimum inhibitory concentration. They knew what happened when the oil touched the wound. They knew what did not happen afterward. 2,500 years of that knowledge was not overturned by the antibiotic era. It was economically displaced by it. The bacterium that is killing people in hospitals right now because it is resistant to every available antibiotic does not know carvacrol is a supplement. Its cell membrane does not have a regulatory opinion on the compound approaching it. The chemistry does not check the patent status of the molecule before it works. A 15 milliliter bottle of wild Greek oregano oil costs $2 per application. The MRSA infection that requires intravenous vancomycin, the antibiotic of last resort, costs $30,000 per treatment course. The cost is immense, and sometimes the infection cannot be resolved at any price. The gap between those two numbers is not a scientific gap. It is a regulatory and economic gap that the pharmaceutical industry maintains. The knowledge is in the journals. The plant is on the hillside. The oil is on the shelf. If this vault changed how you look at the wound on your hand, or the infection your doctor says is antibiotic resistant, subscribe. Share this with someone who deserves to know that the mountain herb that kept ancient armies fighting is still growing on the same hillsides. It is still producing the same molecule and still waiting to be remembered. The next vault opens soon, and what is inside is the one technique every chemical remediation company worked hardest to hide. They want to make sure you never plant this in your backyard.